Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science Podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com. And this week, I'm so happy, delighted, as always. We always have a great lineup of guests, and today is no different. Today, we have with us Dr. Sharon Rosenzweig-Lipson, who is the Chief Scientific Officer at Life Biosciences. Life Biosciences is a biotech company advancing a cellular rejuvenation gene therapy to treat age-related diseases at their root cause. The company is preparing to launch the first ever clinical trial of partial epigenetic reprogramming, targeting eye diseases like glaucoma and non-arteritic anterior ischemic optic neuropathy, or NAION, early next year. Dr. Rosenzweig-Lipson has held leadership positions in both big pharma and small biotech companies over the course of her over 3-decade-long career, which has included roles at American Cyanamid, which was acquired by American Home Products, renamed itself Wyeth, and acquired by Pfizer. She spent her time advancing critical programs from the early exploratory phase through to Phase IIB. Afterwards, she spent the next 12 years at AgeneBio, where she served as VP of R&D. Dr. Rosenzweig-Lipson is globally recognized for her scientific and strategic leadership, having led global translational alliances. She received her Bachelor of Arts in Biological Basis of Behavior from the University of Pennsylvania and her PhD in Psychology and Behavioral Neuroscience from Harvard University. To learn more about the science of aging, methods to reverse cellular aging, and Life Biosciences' work in advancing a first-of-its-kind therapy into the clinic, and to learn more about Dr. Sharon Rosenzweig-Lipson's very foundational and very fundamental work in the field, let's turn to her now. Thank you so much for being on the show with us today.
Oh, thank you so much for having me on the show. I'm excited to be here and to talk about Life Biosciences and what we're doing.
Yeah, likewise. So happy to have you here and so excited to learn more about your career and to learn more about the very exciting work that you're leading in the space of aging and regenerative medicine and all the work that you're helming at Life Biosciences. So we'll get right into it. I'd like to learn more about how things began for you and what piqued your interest in aging research and what led you to Life Biosciences.
So thank you for inviting me here and for the opportunity to talk about this. So my interest in aging actually started all the way back when I was an undergrad in college and started to do my senior thesis and started to work on Alzheimer's disease. So it was really my first foray into science and science of aging and really understanding what the mechanistic differences were. At that time, I was studying muscarinic M1 and M2 receptors. By the time I was done, there were 5 muscarinic receptors. But basically, that was the beginning. I then spent, as you mentioned, a nice career in big pharma, where I had the opportunity to work on both neurology and psychiatry programs all the way through and into the clinic. So that was really the next opportunity to really learn and understand about aging and aging biology. But really at a time where the focus was really on specifics, what goes wrong in the disease and not necessarily what goes wrong in aging. When I moved to AgeneBio, there was the first time where it was really about the science of aging. What's changing in aging? And so a lot of the foundational work at AgeneBio was around aging biology in rats and how you could determine that rats were age impaired or age unimpaired. And by understanding the differences in the biology, you could design therapeutics. And so I led a team there all the way through a Phase IIB clinical trial in mild cognitive impairment due to Alzheimer's disease. All of that was very disease focused. And really where things, changed is when I had this opportunity to come to Life Biosciences. So I've known the CEO of Life Biosciences for many years, and he reached out to me and said, "Hey, want to consult?" And I said, "Well, I'm kind of busy now. I'm doing this Phase IIB clinical trial." And so he'd call me again every couple of weeks until I said, okay, I'll consult. And I had this great opportunity at Life Bio to look under the hood, to learn about the work that was being done there, the science, the biology of aging that's really transformational. We'll talk more about that. Got super excited about it and joined as CSO about a year later. So I've been CSO at Life Bio now for just over 2.5 years.
Wonderful. And I really love that journey where it's kind of like, I'm kind of busy with the, my trials here, but Life Biosciences was indeed pulling you in, because I think definitely for all that you do and for really driving the company ahead, I think they really needed your expertise. And so I'd love to learn more about Life Biosciences and what it's particularly focused on, what areas, what type of biology within aging research, and all of that great stuff.
Yep. So Life Biosciences is a small biotech company. We are focused on very specifically on the underlying biology of aging. And in particular, we focus around cellular reprogramming, or what we refer to as partial epigenetic reprogramming. And this is based on foundational work from both Shinya Yamanaka, who identified these 4 transcription factors, that was built on by David Sinclair at Harvard. And David is the founder of Life Biosciences, and it's really based on the work that he did at Harvard and with his colleagues that became the foundation that we brought into Life Biosciences. And we could talk more about that.
Absolutely. And in terms of aging research and aging biology today, where do you think the field is in terms of, there's so many things, of course, epigenetic, as you mentioned, the epigenetics of aging, which is what Life Biosciences is focused on. And I do remember when I was in grad school, doing my master's a long time ago, I was working on a compound that was initially also studied by Dr. Sinclair in resveratrol in extending lifespan in rats and in mice. And then I was looking at that in oncology as a potential radiation sensitizer. But it was very interesting how really things were taking off then, and really from the biology aspect of things. And the other thing is that, like, when we think about aging research, everyone just wants to find the foundation of the fountain of youth. I'm sorry, but that's not really what it's about. It's about really understanding, like, our cells at the cellular level, the disease, the pathologies that are associated with aging. So, I guess my question is, in terms of today, how far have we come in the last several years and the last couple of decades since you've been in this space, and what is our understanding today?
So I think the aging biology space has really exploded over the last, dozen years or so. First with, the ideas and building out the hallmarks of aging, and really giving us testable hypotheses now across a number of different ways to look at aging and aging biology. That really brought it, very much into a very scientifically focused domain, and really allowed us to sort of test each of these different arenas. The one that's piqued our interest at Life Biosciences is around partial epigenetic reprogramming, because we really think it is a foundational and transformational technology that allows us not just to take aged, mature, injured cells and make them younger and healthier in one tissue, but the same biology can be used and transcend organs and can be used across tissues to do that.
Indeed. Wonderful. And in terms of partial epigenetic reprogramming, could you tell us a bit more about this approach and what it entails?
Yeah, so partial epigenetic reprogramming is built off this foundation, as I mentioned, by Shinya Yamanaka, who identified that you could take 4 transcription factors, OCT4, SOX2, KLF4, and CMYC, and you could take a mature cell and turn it all the way back into an induced pluripotent stem cell. Really cool for stem cell research. Not so good when you want to give it in vivo. So it turns out when you give it in vivo, all 4 factors are starting to take all of your cells and turning them into stem cells. And so you wind up creating teratomas, tumors, things that you don't want. You can very briefly and very transiently administer them and see benefits, but if you give them for more than a few days, they cause toxicity. What David Sinclair identified was you could use only 3 of those factors. You could use OCT4, SOX2, and KLF4. And instead of going all the way back to pluripotency, what you could do is you could take an aged, mature, injured cell and turn it into a younger, healthier cell. Okay, and what's happening here is that we have this ability to reset the epigenetic code. So let's just talk about what that means for a minute. So we know that we take cells from adults and we make babies, right? So what happens when in the zygote stage is that all the epigenetic marks are stripped. So we take all of that all that aging that's occurring. So these epigenetic marks accumulate with age. And the first thing we do is we strip it all, and then we start to add them back in organ-specific ways. This way we can make a liver, a heart, etc. So we create the right code for that we carry into adulthood. As we age, that starts to drift. The code gets disrupted a little bit. The epigenetic code gets disrupted. And in doing so, we start to create cells that are unhealthy in our organs, allowing for disease to start to progress. To the extent to which we can now revert that, restore the epigenetic code just back to the where it was when the organ was young and healthy, we have the ability to reverse a disease and protect against aging.
Wonderful. Thank you so much for explaining that, the approach, and really in terms of now when we talk about diseases like glaucoma and NAION, really, why is this approach, partial epigenetic reprogramming, potentially very beneficial or could have success in these particular indications?
So the eye is a great place to start. So some of the foundational work was done in the eye. We have the ability to deliver OSK easily to the eye. We can just do it in a single intravitreal injection. It's a common approach to treating eye diseases. So in macular degeneration, you get intravitreal injections every few weeks. So this is a one-time intravitreal injection. The eye is a bit immune privileged, so we can put an AAV-based therapy into the eye without having as much of an immune response as you might have in other organs. But the other thing is we pick the eye because if we want to talk about healthy aging, so we're not necessarily talking about, extending lifespan, but really we want to take that period of time where rather than there's a long period of decline and sickness before you pass, but rather to take that period and make that healthier. Well, to make aging healthier, we all lose, not we all, but many people are losing vision. It's one of the single largest issues in aging. And we know that as we start to lose vision, we lose our ability to live independently, work independently. We're at greater risk for falls, bumping into furniture. We're at much greater risk of injury. It also starts to socially isolate us and leads to mental health issues and cognitive decline. So to the extent to which we can protect against vision loss or reverse vision loss, we have this incredible opportunity to improve healthy aging. And so that's where we decided to go there first.
Yeah, that's very insightful, really, to learn more about why the eye is such a great target for epigenetic reprogramming. And as you mentioned, really the quality of life deteriorates, with eye issues, and it affects just so many things, mobility and cognition, and the eyes are definitely the window into the soul and everything else, I think, in the body. So absolutely. And in terms of developing these kinds of therapies, what are some of the biggest scientific or clinical challenges in the space right now?
So I think, with respect to what we're doing, safety first. So I mentioned that the 4 factors create some risk. With the 3 factors, we don't see those risks. We can maintain cell identity. We don't induce tumors or teratomas, but we had to confirm all of that. Um, and then we really wanted to build a very translational program. So we wanted to demonstrate first in mice that you could improve in a number of different models of retinal ganglion cell dysfunction. So that's all these optic neuropathies. Glaucoma and NAION are both disorders of retinal ganglion cells. And so we wanted to demonstrate across a number of models in mice. But then we had to just take the next step, which is mouse vision is not the same as human vision. They don't have a fovea, they don't have macula. And so now we wanted to do is take it to the next step and say, could we do this in a non-human primate? Could we demonstrate the same benefits that we see in mice that we could transcend? So we used a model of NAION in non-human primates, and we were able to demonstrate these improvements. So taking all of that and then building on the safety profile, what we did was one other thing. We created a inducible system. What that means is when we give the injection of ER100 into the eye, OSK will not be expressed unless we also give patients doxycycline, okay? Just a commonly used antibiotic. But that commonly used antibiotic turns on the expression of OSK. It also means we can turn off the expression of OSK. So we can give it for the period of time we think we need, and then we can just remove the doxycycline and it's off. That allows us another layer of safety control in our trial. And then, moving into this whole area of cellular reprogramming, we believe we're the first that'll be moving in partial epigenetic reprogramming. So identifying the regulatory pathway and all of the steps that are needed to make sure that we can be successful were quite important. So all of these are important steps. And now we're very excited because we're about to go into the clinic. We've crossed all those boxes, and we intend to be in the clinic in the first quarter of 26.
Fantastic. And looking at, I guess, the broader field, what are, of course, you touched on the very critical unmet needs, but what other things are really in the space in indications and diseases like glaucoma and NAION that you're really hoping to target and achieve with epigenetic-based therapies?
So the really exciting thing about epigenetics and what we're doing is that epigenetics is involved in aging across all the organ systems, and a wide array of diseases. I like to tell people, let's play head, shoulders, knees, and toes. But basically, you can go, from head to toes, and there are epigenetic changes that are occurring with aging and diseases of aging as a consequence of that. So the exciting part is that we have this ability to do it across diseases. We just presented a few weeks ago at the age-related drug discovery meeting that not only can we do it in the eye, but we decided let's try and find a cell type that is so different from the eye. So we decided to go after the liver, and we were able to demonstrate improvements using OSK in a mouse model of liver disease or metabolic associated steatohepatitis or MASH. So we're very excited that we have this next indication where we've been able to demonstrate. But what we really did was we transcended the technology and really transformed where we can go. We can go not to any organ. And it really, there's so much opportunity, and we've got a long list of where we want to go next.
That's so cool. Dare I say, but it's like absolutely like potentially being able to target so many organ systems and really, really fascinating stuff. And
right. Another really cool thing about this technology is it knows what to do, so to speak. So what I mean by that is if we're saying that we're going to do partial epigenetic reprogramming, we're saying we're going to change the DNA methylation profile of methyls on the DNA. So in neurons, it changes the methylation profile on genes involved with neurons. In other organs, it doesn't change neuronal. Remember, we have the same genes in every cell, but it doesn't change neuronal genes. It changes, for example, in the liver, hepatic genes. Okay, and so in each case, it's resetting the code back to what that organ was supposed to be. And that's just a really super cool component of what this technology can do.
A hard reset is something that I think we all need from time to time, dare I say. The devices we use and ourselves too, to take a step back and to reset.
We all modify our epigenetics every single day based on what we eat, based on what we drink, based on whether we exercise, whether we're smoking. All of those things have the ability to change our epigenetics and change our aging profile. And then to the extent to which we're accelerating the aging of any one organ, we're increasing the risk of an age-related disease in that organ.
Yep. And so, a fix to reset things definitely sounds like a great approach indeed. And in terms of when we talk about just, I think I alluded to this earlier, but when we talk about the field of aging and we talk about just aging in general, obviously in this context here, when we're talking in the life sciences, drug development for us, it's like we're obviously thinking the pathophysiology is associated with cellular aging and all of those things in terms of diseases. But what are some common misconceptions, even maybe perhaps for people in the field and also for obviously the lay person, about aging and disease?
So I think there are a couple of things that, often happen. If you go into a room full of people and say, raise your hand if you want to live forever. Okay. Most people won't raise their hand for that. Okay. If you ask that same room and say, if you could live longer and healthier, okay, and you could maintain your current health, would you want to continue? And everybody raises their hand immediately, right? Because if I could stay like I am or stay younger and healthier, immediately, if you say live forever, you think of aging, sickness, illness, and it's a pathway that's scary. So I think, one of the misconceptions is that we're really not about living forever, but really what we want to do is improve health span that we think ultimately may improve lifespan as well.
Absolutely. Wonderfully and very, aptly put, for sure. I don't think people are looking for immortality, but really a healthy lifespan and life. Very key.
Right. When you want to shorten that period of decline,
Yeah.
we don't want this long period of decline.
Have a prolonged,
exactly. We want a long period of healthy aging.
Not a prolonged period of age-related decline and all of that.
Age-related diseases and illness and, things that are affecting quality of life.
Yeah. And in terms of looking at the drug development process and just drug development in general, we talk about we're seeing a lot more partnerships and collaborations in general, whether it's between big pharma, smaller biotechs, with academia and industry in general as well. So we're seeing a lot of those kinds of partnerships. And so from your perspective, having been in big pharma and now at a smaller biotech and having seen really the spectrum of it all, what really do you see today happening in this space in terms of partnerships? And also, what would be the pros and cons between big and small pharma and all of those elements?
So I think partnerships are critical and important. We're actually a relatively virtual company. We don't have our own lab. So we do a lot of our work through partnering. So we're partnering all over the world, wherever the best research is being done, where the best models are, where the best manufacturing is. So we've, partnered, for example, with Forge to build our manufacturing capabilities. We recently signed a memorandum of understanding with Remedis in Singapore, or I think it's regenerative medicine in Singapore, to do some work there. But really, so we're doing work around the world wherever it's, best in class. As far as partnerships with, big pharma and biotech, I think they're all important. I think biotech can take it just so far. At some point, the financial needs just become beyond what biotech can manage. And so often, that's where big pharma partnerships are really important. And big pharma brings a lot of expertise to the table. So there's really valuable interaction. So I think you see a lot of that. And big pharma is, doing less sometimes of their own drug discovery, allowing the small biotechs to do it. And then they get to pick, what they think is best.
Oh, sorry, I was just finding the mute button there. And in terms of really, if you look back on your journey and looking forward as well, really leading in the space, leading in innovating and in drug development, and given your experience and your insights, what can you glean from them, and what would you advise emerging leaders in the space, and basically what advice would you want to give them in terms of if they're looking at entrepreneurship, whether starting up their own biotech or coming on to as a CSO somewhere? What should they be looking out for today?
So I think the advice I like to give to everybody is follow your passion. Where are you interested? And keep following it. And take opportunities when they come. Don't be afraid of them. Often people look at a new opportunity, they go, well, I don't know, I haven't done it. I'm sure I could. Yeah, I probably could do it. Instead of just embracing it and saying, there's a new challenge, there's a new opportunity. When you go from big pharma where you're sort of siloed into an area of expertise a little bit, into biotech where you have to know a lot more about everything that's being done. So don't be afraid to take that on. Use it as an opportunity to learn. In every step of my career, that's what I've done. I've taken and said, what can I do to expand what I'm doing? While I was at big pharma, I had this great opportunity to take my program from early exploratory and stay with it all the way into phase 2 clinical trials. Well, that gave me a really breadth of insight into the whole drug discovery process, in that atmosphere. Going to biotech, now I became, an amateur formulation, person, and I became amateur in a bunch of different things. But really, it's about learning the language, learning what they're doing, learning why it needs to be done. I've discovered often it's all just data. As a scientist, it's data and it's interpreting data. So as long as you sort of get an understanding of the system and what kind of information you need, you can troubleshoot anything. And so really believing in yourself and following your passion to wherever it takes you.
I love that. And in terms of Life Biosciences, what milestones are you looking forward to the most, and what are you most excited about? I know that you mentioned that your lead candidate is ready to head into the clinic, so definitely that's truly very exciting. And so
number one is file the IND, and number two is start the clinical trial. So that's obviously the most exciting. But I think the other thing is now that we've transcended from the eye to the liver, it's really, where else can we go? What else can we do? And so we've got more proof of concepts in the books, and that we're working on that, we hope to be able to share soon.
Wonderful. And what excites you most about the field moving forward, having seen the field evolve, and you having been really within that trajectory and within that journey? So how have you seen the field evolve, and where do you see it heading in the next couple of years? What kinds of technologies, approaches are you most excited about?
So, within the aging field, I think just this array of technologies that are maturing right now to the point where we can start testing them all clinically, is very exciting. And so, seeing where all of that goes. Obviously, the field of partial epigenetic reprogramming has been exploding, and there are, number of companies doing it. So we think there's so much opportunity for all of us to succeed and to grow in the field. You could think of this like the early days of cancer research, and then, watching it explode with lots of companies and lots of opportunity. We think the same is going to be true for partial epigenetic reprogramming. And we're thrilled that we are, believe we'll be the first into the clinic with it.
Fantastic. And when we talk about different technologies, AI, of course, comes to mind. And, is it hype? Is it a bubble? We don't really know yet. We'll know in a couple of years for sure. But leveraging these kinds of technologies, digital tools, biomarkers as well, what really stands out most in aging research in terms of utility to you?
So I think there too, I think AI transcends. It doesn't limit itself to aging biology. It's, to drug discovery as a whole, and well beyond outside the science arenas. And super exciting to see where AI is going to take us and whether we can really both expand the targets that we're looking at, expand how we do chemistry, how we run clinical trials, how we bring in patients. There's so many different ways how we identify patients. All of that, AI will be potentially transformative. The biomarkers, they're so critical, as we move into aging and really understanding the biomarkers of aging. And there's so much work being done around different clocks that can be used that are showing you different epigenetic changes or other biological changes that really help you to find what's actually being modified by aging and whether or not we can reverse them. And as we move into the clinical arena, having these biomarkers ready for clinical trial use is going to be critical.
Dr. Rosenzweig-Lipson, thank you so much for your time today and sharing your insights and all of the amazing work that you have done and are continuing to lead in the field. And we'll definitely keep an eye out for all of those milestones coming up at Life Biosciences, and wish you all the best. And thank you for all of the work that you do.
Thank you so much for having me. It's been so much fun talking to you.
Likewise. All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next time. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.